All right, so this is just another example or another display of the figure that we basically just looked at a moment ago. Um, but it's worthwhile to kind of have another perspective and another artist rendering basically of what all this looks like. We're going to dive into some of these things a little bit more in just a moment. But one of the things that's important to remember, important to know about this, is that integral proteins are associated very tightly. So we already talked about an integral protein as something that spans that membrane. These are like locked in, they are bought in there, definitely a part of this membrane. And in order to displace that, you're going to have a detergent to break up a membrane or a chaotropic agent that's actually going to help disrupt those hydrogen bonds in order to separate that from that membrane. I remember when I was in graduate school, we had one professor who said, oh, if you're studying a membrane protein or an integral membrane protein, go ahead and add a year to your PhD studies. And so the reason for that is just the logistical nature of getting that protein out of that membrane and not destroying the protein in the process because they're very tightly associated. Um, Lipid linkages tend to be globular after their release. So lipid linked proteins, these guys, so the one right down here is an example of a lipid linked protein that's probably going to be globular whenever it's actually disrupted from that linkage to that lipid. Okay, now peripheral proteins as well. We saw in the previous slide where a peripheral protein is it's in contact with and associated with, but you don't have any sort of covalent linkage that keeps that protein in there. So basically membrane proteins fall into three major categories. Your peripheral proteins that they're there, they're hanging out, they like the membrane, but they're not living there or anything. Lipid linked proteins where you have a little bit more of a static interaction, or you have, yeah, a little bit more of a static interaction because you have a covalent linkage, a post-translational modification to that protein that associates it with the membrane. And then finally, your third group is your integral proteins, which are integrated into that membrane and are associated with that membrane. Okay, so when it comes to these integrated proteins or integral proteins, um, they fall into a number of different categories. So what I'd like to, again, draw your attention to very first is what constitutes the inside and what constitutes the outside of our membrane here. Well, these proteins fall into a total of 1, 2, 3, 4, 5, 6 categories. Okay, so type 1 and type 2 are basically the same, but what about the orientation of those, the N termini and C termini? So your N termini is going to be on the interior, the interior of the cell, or interior of the membrane on a type 2 protein. So again, N termini on the inside in a type 2 protein. A type 1 protein, I'm sorry, your N termini is going to be on the outside. Type 3 and type 4, they look fairly similar to one another. The orientation of the N termini versus the C termini is really not quite so important, but really what's more important is the alpha helices. So with type 3 and type 4, we have one single polypeptide. Whereas type 4, you have multiple polypeptides. So they're kind of grouped together in that they're multiple helices spanning the membrane. However, one of them is the result of one polypeptide and the other is the result of multiple polypeptides. So when it comes to tertiary and quaternary structure, well, that type 4 is going to have a quaternary structure because it's multiple proteins associated with one another. Type 3, however, is only going to have a tertiary structure. Type 5 protein is a great example, or type 5 transmembrane protein is a great example of a lipid-linked protein because this protein is found on the inside of the cell, and there's this small molecule, this small black molecule right here, that's the lipid linkage that gets it to associate with the membrane. Type 6, on the other hand, is an integral membrane protein that both spans the membrane and has a linkage on the exterior of that membrane that causes it to further associate with it. So this one is kind of all bought in, and it makes sure, and we've got some sort of anchor of a portion of the polypeptide chain that keeps it in close proximity to the exterior of membrane. Now over here, what I'd like to draw your attention to is this shows a polypeptide chain, and one thing that you can do is look at this and say, is this a type 1 or a type 2? We've got an alpha helix right here that spans the membrane. Our N termini, our amino terminus, is on the outside of the membrane, and our C termini is on the inside. Okay, so if our C termini is on the inside, that means that this is a type 1 integral membrane protein. Now, if you could zoom in for a second on each one of these, and I'm going to try, I believe that I can see that. As it turns out, I actually cannot zoom in on this picture, but what I'd really like you to focus in on is the amino acids that are found within our lipid bilayer. So the residues that are within this protein that come in contact with the hydrophobic tails of all of our fatty acids within that bilayer, well, many of those are highly hydrophobic residues. For instance, I see a lot of glycines and valines. Those are residues that they're generally not going to hydrogen bond with anything, and in fact, they're going to be fairly nonpolar. Okay, so that's one thing that's worth noting about these different types of transmembrane proteins and these residues.